Hello, my name is Marc. Now today we're in the Ecuadorian cloud forest on the edge of the Zunag River, and today I'm going to go hunting for microscopic life in the pools around the river here. And then what we will do, we'll take it back to the lab and have a look at it under the microscope. So let's go. Right, so this little pool is just a little bit away from the river. The water's quite stagnant, and in here it is absolutely chockers full of tadpoles. There's loads and loads of small ones, and there's also quite a few big ones here as well. And at the bottom, it's nice and green. It's got like lots of pond scum in there, which means there's lots of life, lots of stuff that the tadpoles are feeding on. So let's get in a bit closer and scoop some of this stuff up. I've got here a little syringe just to suck stuff up from the very bottom. So let's get in there and suck some of that green stuff up. And I might even try and get some couple of little tadpoles if we can. Yeah, just one will do. Scrape some slime off as well and get a hole. Let's just use our fingers, why not? Get in there, a bit of dirt. So there we go, we have our sample. Bit of a mess in there, there's a couple of tadpoles and just a lot of goop down in the bottom. Plenty of green stuff. And let's take that back to the lab and have a closer look. This is the microscope that we're going to use. It's an Olympus KHS. It's quite old, but it's still got some really great glass here in these objectives. And I've modified it so I can attach my digital camera on the top here. For starters, let's see if you can guess what this one is. This is using the microscope with a 40 times objective and almost the maximum magnification that I can do without using a special oil lens. And this is zoomed out a bit more using the 10 times objective, and this is zoomed out again using the 4 times objective. Now, if you haven't guessed what it is, it's actually the tail of the tadpole, and what you were looking at before was the actual blood cells moving through the vessels in the tail. Now we'll move over to where the gills are and we'll zoom in on the actual gills. You can see there's a bit of movement around the gills, and the gills, they're not like a standard fish's gills, they're just protrusions from the tadpole's body. And if we zoom in again, we can see the tiny little cilia that help to move the water around the surface of the tadpole's gills. And this is where the actual exchange of oxygen and carbon dioxide happens with the surrounding water. Now that tadpole was about 3 millimeters long. Now these guys are about 0.1 millimeters long. It is called Paramecium and it is a Ciliate Protozoa. One thing I like about using a microscope is that it gives you X-ray vision. This is because the light you're observing is passing through the organism, as opposed to what you would normally see is reflected light. Like the tadpole gills, Paramecium is covered in cilia, and it uses these tiny hairs like thousands of tiny oars to propel itself through the water. The small strands that you see moving back and forth are bacteria, and these are so small that they approach the wavelength of visible light. This makes them very difficult to observe in detail under a conventional optical microscope. I found this Paramecium which was stationary, and then I was lucky enough to observe this. Now whether the Paramecium is dying and has some sort of bacterial infection, or if it was actually reproducing, I was not 100% sure, as I was not able to observe the end of this process. If you have any ideas, let me know in the comments below. This is another type of protozoa, an amoeba. It doesn't have cilia and it moves by oozing itself through the water. Peranema is another protozoa that also oozes through the water, but it has a flagella on its front, which it looks like it's using it to sense its environment, and it also can use this to pull itself along through the water. If you look very, very closely, you can see a small rod-like structure in what might be called the head of Peranema. Peranema can extend and retract this small rod, and the theory has it that it can use it to batter a hole in the lining of its prey organism. It can then use its flagella to mix up the insides while it actually sucks those insides out. Now, if I'd known this at the time of filming, I probably would have spent hours hunting around and following this guy to actually try and film this behaviour. This is a diatom. It is a single-celled algae. Relatives of these in the oceans are responsible for an estimated 45% of all organic production. This is another form of algae, Spirogyra. This is a multicellular algae whose individual cells attach together to form long strands. The outer surface of these cells is covered in pectin, which is similar to what you might find in your typical supermarket jams. This mixes with the water and makes Spirogyra slimy to the touch. And in large enough quantities, you get natural green slime. This shot is actually a time lapse, and if I slow it down just for a moment, you can see that relative to Paramecium, the algae does not move at all. Likewise, the movement of this Filamentous bacteria is also only visible under time lapse. Under maximum magnification and in real time, you can just make out the transport mechanisms moving nutrients inside the Spirogyra cells. You can also see the individual green chloroplasts. Chloroplasts contain chlorophyll, which allows plants and algae to convert sunlight into energy. This is the main predator that I found in the sample. It is the larvae of a midge fly, and currently this is being played back at half speed because he really does move fast. Let's return to full speed and have a look at him. This is his rear end or tail, and this is his head and his forepaws. Now, whether he's moving this fast because he is hunting, or he's just moving like this because he's stuck between two bits of glass, I'll leave up to you. Either way, he's definitely aggressive, and I'd say he'd be preying on those little Paramecium and anything else that he can get his jaws around. Let's look at some stills so we can get a clearer look. He's got two probably touch-sensitive feelers, claws on his forepaws for gripping the Spirogyra, and claws on his rear end as well. These are retractable so he can move his rear end forwards with his very flexible body. And look at those jaws, definitely made for killing. Thank you for watching my video. If you have enjoyed this, then please subscribe below and please give the video a like and throw some comments in there. It all helps. Now, I have a Twitter account as well. I sometimes upload photos and stuff of the work that I'm doing at the time, and I also have a couple of other YouTube channels. So when I upload videos to those and this channel, they will appear on my Twitter account as well if you want to follow me that way. I have a website, On that website, there's various information on the gear that I use. So if you're interested in that, please go and have a look. There's a few affiliate links in there as well, which helps to support my work. I'll leave some links to some of my previous videos here. And if you ever want to search for my videos on Google, all you need to do is put in Biodiversity Shorts and then hash and the number of the video that you want to watch, and that will come straight up on Google. Anyway, thanks again. I'll catch you next time.